Ion Torrent is a next-generation sequencing technology that was developed by Life Technologies in 2010. While it is similar to most NGS platforms, it differs in that it doesn't use fluorescence to call bases. Instead, it detects hydrogen ions released upon base incorporation and converts it into sequencing information.
To give an overview, the ion torrent process consists of hybridizing your DNA fragments to microscopic beads, and then flowing the beads into individual wells on a semiconductor chip. Individual nucleotides are then incorporated one at a time, and the hydrogen atoms released upon base incorporation are interpreted by the machine as a base call.
The first step of the process is library preparation. To start out, you need to fragment your DNA into uniform sizes. You then ligate sequencing adapters to both ends of the DNA fragments. These adapters are complementary to the primers that you will coat your beads with. You then incubate your DNA with the beads to allow for hybridization of your DNA fragments to the bead surfaces. It is important to use proper dilutions of the fragment solution to ensure that each bead only receives a single fragment of DNA. The next step is using emulsion PCR to coat each bead with millions of copies of the original DNA fragment. Now that you have your beads coated with the DNA, the next step is flowing them onto a specialized semiconductor chip. This chip is similar to those found in digital cameras, but instead of transmitting photons into digital information, this chip detects changes in local pH and interprets it as sequencing information. Once you have your beads all contained within single wells on the plate, a solution of one of the 4 nucleotides is flowed over the chip one at a time. If the base is incorporated by DNA polymerase, a hydrogen ion is released into the solution of the well, raising the local pH. A special ion-detecting layer underneath the well interprets this change in pH as a base call. This process continues one nucleotide at a time until all of the fragments have been sequenced.
So how does Ion Torrent compare to Illumina? Basic read lengths for Illumina are 150 to 300 base pairs, while Ion Torrent has a slightly larger range based on the size of the total sequence. For larger sequences, read lengths can be as small as 100 base pairs, but for smaller sequences, read lengths can be up to 400 base pairs. For overall application, both Illumina and Ion Torrent can sequence RNA or DNA, and everything from targeted sequences to a small genome. Currently, only Illumina can sequence a whole human genome. This is mostly due to the capacity. Illumina can sequence 25 million to 6 billion single reads, whereas the Ion Torrent has a maximum of 330 million single reads. A major benefit to using Ion Torrent, it is much more cost-effective compared to Illumina. The most expensive Ion Torrent sequence is still less expensive than Illumina sequencing. Ion Torrent is also incredibly fast compared to Illumina. Sequencing can take anywhere from 26 hours to 11 days, whereas Ion Torrent can be prepared, sequenced, and processed within 24 hours. As for the error rate, per the Illumina sequencing website, the error rate fluctuates from 0.1% to 10%, but the Ion Torrent is much more test-dependent and will be explained in more detail.
If there is a chain of homopolymers in the DNA fragment, the entire string of the identical bases will be incorporated within the same round of nucleotide addition. The change in pH caused by the incorporation of the homopolymers is not necessarily proportional to the number of bases added. If there are greater than 5 of the same base, the ion detecting layer has a difficult time distinguishing the number of bases included. When there are fewer than 5 base pairs added, the peak in pH should be almost proportional to the number of bases added. In order to combat this issue, the use of optimal flow order can help mitigate the error rate. This is something to consider when using Ion Torrent.
DNA sequencing used to be impractical and expensive. However, Ion Torrent has made DNA sequencing cost and time effective. Ion Torrent has many different applications, but one specific application is that it can be used to identify cancer mutations. In a paper that was investigating gene mutations in human breast cancer, Ion Torrent was used to sequence over 700 different loci from 45 genes that are cancer-related. Using the Ion Torrent, they could identify genetic mutations in 105 breast cancer samples. Missense mutations were observed in 2 different genes, and this information can now be used for treatment development. Ion Torrent is one of the fastest benchtop next-generation sequencers with many applications. It can be used for targeted sequencing, microbial sequencing, de novo sequencing, small RNA and microRNA sequencing, and much more. The development of ion Torrent sequencing has been beneficial in many different fields of science and is continually improved.